Hello, everyone, and welcome to today's live webinar, Automated Multiplex Immunofluorescence, presented by Dr. Bethany Remeniuk and Traci DeGeer. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Biosystems. For more information on our sponsor, please visit the Sponsor tab at the left side of your screen and click on the logos to be directed to the sponsor website. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or use the Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to present today's speakers, Dr. Bethany Remeniuk, Global Application Scientist, Akoya Biosciences, and Traci DeGeer, Director, Advanced Staining Innovation, Leica Biosystems. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Bethany and Traci, you may now begin your presentation.
Thank you, Christy, for that introduction. Hello, everyone. My name is Bethany Remeniuk, and I am the Global Application Scientist at Akoya Biosciences. Today, we will be discussing the advantages of utilizing Akoya's Opal Multiplex Immunohistochemistry automated detection kits on the Leica Biosystems BondRx autostainer to meet your research needs. In addition, I will be highlighting some tips and recommendations to help design your multiplex panel and optimize your staining. However, before we can delve into this, we must first take a step back to understand how these two platforms combined help to facilitate a streamlined workflow and drive cancer research forward. From 1991 to 2015, cancer deaths in the United States alone fell by 26%. This is due, in part, to significant advances in the treatment of cancer patients, including the use of more personalized therapies for individuals, as well as FDA-approved immunotherapy treatments such as anti-PD-1 and anti-PD-L1. However, not all individuals who test positive for these markers will respond to targeted treatment. To help understand why we may have responders and non-responders, we must first acknowledge that the biology and composition of the tumor microenvironment is very complex. There are several specialized cell types, including immune-mediated cells, such as macrophages and B and T cell lymphocytes, that interact directly and indirectly with tumor cells, resulting in an ever-evolving relationship between immune cells trying to destroy the tumor cells, while simultaneously the tumor cells attempting to evade and promote their own self-growth. Because of this, there are several influencing factors that need to be taken into consideration when interrogating the tumor microenvironment, including cellular phenotypes that could influence the biology of the tumor, the functional status of the surrounding cell types, as well as other contributing factors such as inflammatory mediators and how these all interact within the spatial biology of the tumor microenvironment. Having a greater understanding of the relationships happening within the tumor microenvironment are key to improving diagnosis and designing new therapeutic regimens. Currently, clinical laboratory tests are automated and performed by calibrated machines to reduce human error. However, the majority of anatomic pathology and disease diagnosis is still based upon physician interpretation of microscopic tissue characteristics, such as cells, markers, and tissue architecture. In fact, this visual assessment using conventional immunohistochemistry remains the gold standard for cancer diagnosis. Despite this, it is recognized that visual assessment can be subjective and interpretation of results can differ between pathologists. Additionally, because immunohistochemistry, such as DAB, can only be conducted for a single marker on tissue samples, very little information can be obtained about the spatial context and interplay of other cellular phenotypes within the tumor microenvironment. On the other hand, genomics and proteomic studies, including flow cytometry and DNA microarrays, provide significant quantitative information regarding different cellular phenotypes within the tumor. However, the spatial context is lost as analysis needs to be conducted using homogenized tissue. Thus, we're at this crossroads where on one platform, we're able to retain morphology but lose phenotypic information, or conversely, have a wealth of phenotype data but lose spatial biology and interactions between them. In order to advance research forward, we need to be able to retain the spatial biology of the tumor microenvironment while simultaneously identifying and labeling multiple markers of interest. And this is where immunofluorescence is truly able to bridge the divide. Immunofluorescence overcomes the limitations of conventional immunohistochemistry, which are constrained by detecting 2 to 3 markers of interest, by allowing for multiplexing of several markers. By using this methodology, researchers are able to conserve their tissue samples while identifying and characterizing cellular interactions. Immunofluorescence can then identify and reveal weak expressors that may be lost using conventional immunohistochemistry staining. And furthermore, the balancing of signal can allow researchers to detect multiple markers that co-localize on the same cell. Lastly, immunofluorescence can provide better signal for quantification and data analysis. While there are many benefits associated with immunofluorescence, there are challenges with conventional methodologies that can unfortunately limit your staining success. First, staining will need to be done using antibodies raised in different species, as secondary antibodies may cross-react and ruin your results. This, unfortunately, can be restricting, as some rare antibodies may only be raised in one species. Next, some antigens may be sheltered from the primary antibody due to steric hindrance. Furthermore, signals can be unbalanced or low if you have an overabundance of a marker or very little expression of another. Other factors that may impact your results include background staining, contributing tissue autofluorescence, which can be a result of the tissue itself, as well as how the tissue was processed, and photobleaching or quenching of the fluorophores if they are exposed to light. Thus, the Opal fluorophores were created to overcome the limitations of conventional immunofluorescence. As part of the Phenoptics workflow, Opal dyes are accessible to anyone who has previously worked with standard immunohistochemistry. Opal allows for detection of multiple cellular phenotypes to be visualized and quantified simultaneously in the same formalin-fixed paraffin-embedded tissue section, enabling researchers to study the complex relationships and distribution of these cells while retaining spatial context within the tumor microenvironment. With Opal, you can select any primary antibody you wish for immunohistochemistry detection based upon their performance rather than being relegated to a species type. And lastly, Opal's multiplex assays and kits provide a practical workflow that allows for the simultaneous detection of upwards of 8 biomarkers of interest plus DAPI nuclear counterstain within a single tissue section. Akoya offers Opal automated IHC detection kits for researchers to perform Opal multiplex staining on one of the leading research automated staining platforms, the BondRx by Leica Biosystems. Automation provides you with the flexibility to support the dynamic demands of translational research. The open platform promotes a simplified workflow that frees up users' time for other research endeavors. High throughput Opal bond protocols can perform upwards of 7-color immunofluorescence staining on 30 slides, which can be performed overnight as opposed to typically running manually, which may take 2 to 3 days. And lastly, automation can achieve quality, consistency, and reproducibility with every sample and supports the transition into clinical studies. So how does one actually go about staining with Opal using the BondRx? Well, as previously mentioned, Opal dyes allow for the use of any standard unlabeled primary antibody, including multiple antibodies raised in the same species. Here we have a microscope slide that has been barcoded to be recognized by the BondRx. After introduction of the primary antibody, the Opal polymer HRP is applied. The Opal system then uses tyramide signal amplification, or TSA, to amplify IHC detection by covalently depositing multiple fluorophores near the targeted antigen. After labeling is complete, antibodies are removed in a manner that does not disrupt the Opal fluorescence signal, allowing for the next target to be detected without antibody cross-reactivity. These steps are repeated for all markers of interest. The Opal technology thus enables development of multiplex assays with balanced quantitative signal for both rare and abundant targets of interest. So moving forward, we wanted to test the stability and reproducibility of Opal on the BondRx. First, to confirm fluorescence signal stability of the Opal dyes on the BondRx, we conducted a series of repeated stripping steps with each Opal fluorophore and determined that over the course of 6 repeated high-intensity epitope retrievals, that the Opal dye signal remained stable for all fluors, including our classic 6 fluors that we have, and our latest addition of fluors, including the Opal Polaris 480. Next, to demonstrate Opal's ability to detect markers in the same cellular compartments without interference from TSA deposition, we stained tonsil with both CD3 and CD8, as they are often found in close proximity with one another on the same cell. On the left, we have a merged overlay of the CD3 and CD8 channels, where signal overlap appears in white. And then we have the individual channels appearing to the right. As you can see, TSA does not interfere with the detection of co-localization of markers of interest. Next, we wanted to test the consistency and reproducibility of the BondRx at staining Opal. Here, we ran Monoplexes staining for CD20 using Opal 520 on serial sections of tonsil for all 30 slide positions. We demonstrate that slide variability between all 30 slides for a per cell expression of CD20 produced a coefficient of variation less than 5%, indicating that the bond is able to produce consistent and reliable staining results. For the next testing procedure, we assessed the BondRx's ability to reliably stain 15 tonsil serial sections for a 7-color multiplex panel. For each of the Opal fluorophores, we analyzed the mean counts of the top 20 brightest cells per slide and found that for each marker, there was a calculated CV of 14% or less, which, given the nature of serial sections, speaks to the consistency of both Opal staining as well as the functionality and reliability of the BondRx in producing consistent results for all your staining needs. Next, we're going to delve into some tips, tricks, and recommendations to help optimize your Opal staining. Here on the left, we have an overview of how staining is run on the BondRx using the newest Opal Polaris immunohistochemistry automated detection kit, which includes our newest fluorophores, the Opal Polaris 480 and the Opal Polaris 780. Opal protocols are auto-populated on the BondRx and can be used as they are for all of our other kits that we have. However, if you wish to do staining with the Opal Polaris kit, you will need to modify that protocol to include Opal Polaris 780 at the end of the staining run. And at the end of this presentation as well, I will also share some additional staining resources to help support your staining endeavors. So up first, we have some recommendations from Akoya to help in your monoplex development. First, we recommend that you use tonsil to optimize all of your monoplexes. And this is ideal because tonsil has a number of markers of interest that are highly expressed, making it ideal to help optimize your primary antibody dilution. We recommend going ahead with a series of 3 DAB titrations and then optimizing from there based on staining pattern and intensity. Once you feel you've optimized your primary antibody dilution using DAB, we recommend then converting it to immunofluorescence. Here then you would want to pick an ideal Opal pairing to go with the primary antibody. And some factors to consider include co-expression. So if you happen to be looking at multiple markers of interest on the same cellular compartment, you might want to choose an Opal pairing that for that primary antibody that is perhaps a little bit far removed from the other marker of interest that you are looking to image. For example, you may want to choose Opal 520 paired to CD3, and then maybe Opal 570 or Opal 620 to look at CD8. Furthermore, you also want to take into consideration whether or not you are investigating rare versus abundant markers. If you are looking at markers that may not be highly expressed or weak expressors, you may want to pair them with an Opal that is very bright. In this case, Opal Polaris 480 or Opal 520. Versus if you happen to have a abundant marker of interest, such as cytokeratin, you may want to pair that with some of the more lower bright Opal dyes, including Opal 690 and Opal Polaris 780. For Opal concentrations, we recommend starting at 1 to 150 dilution and then titrating up or down as needed, because we like you to remain within the already predetermined primary antibody dilution. Next, you'll want to go ahead and assess your target brightness, and we recommend that target brightness counts fall within a range of 10 to 30. With the exception of Opal Polaris 780, because it is near the infrared, we recommend that you target anywhere from a range of 1 to 10. This can be done using our one platform, which is free. It's PhenoChart, and you can go ahead and then look at the normalized counts, and then you can adjust your Opal titration from there. Once you get done optimizing your tonsil monoplexes, we recommend transitioning those dilution factors onto project tissue samples and re-optimizing as needed. And again, we recommend not really adjusting the primary antibody dilution factor and trying to titrate out your Opal. Next, for each project, Akoya recommends that you create a new library slide set to ensure you obtain the best fluorophore spectrum to extract from mixing if you are using a Phenoptics imaging system. To achieve the best results, we recommend using tonsil once again and staining for CD20, as it is ubiquitous, uniform, and it is highly expressing. You will want to stain one slide per each fluorophore, along with one slide for DAPI. In addition, you will want to create an autofluorescence slide, but this you will want to use tissue that is representative of your project, as autofluorescence is not the same amongst tissue types, and using tonsil for this may not reflect the amount of autofluorescence that your project tissue samples may have. Lastly, once you have optimized your monoplexes, it is time to pull everything together and stain in a multiplex panel. Here, you want to consider, in particular, staining order. When staining, you want to take into consideration antigen retrieval, as certain antigens can become more exposed after repeated rounds of antigen retrieval, and these include PD-L1 and Ki-67, which you may want to have later in your staining order. Conversely, there are other antigens that you may want to put at the beginning of your staining order because they may become more degraded as you have repeated exposure to antigen retrieval. Next, you also want to take into consideration the biology as well. Some fluorophore intensity can be attenuated after multiple antigen retrievals, including Opal 520 and Opal 570. But as you saw on the earlier slide, overall, though, signal intensity remains consistent throughout the staining, but it is something to take into consideration. Lastly, for image acquisition, you'll want to go ahead and consult the instrument handbook or user manual that came with your respective platform when wanting to image your multiplex panel. And as I mentioned earlier, for more information pertaining to designing and optimize your Opal staining on the Leica Biosystems BondRx, please use the Opal Multiplex IHC Assay Development Guide, which can be found on akoyabio.com. There will actually be a new updated version coming out in the next few months, so be sure to keep an eye out for it when that does happen. In addition, under the resources section of our website, we also provide free Opal Multiplex automated protocols that utilize the Leica BondRx. And then lastly, our European Applications and Development Leader, Virginie Goubert, gave a great online tutorial that delves step-by-step into the Opal Multiplex IHC optimization process. So if you have any other questions, please feel free to go ahead and utilize these resources. In summary, we have demonstrated that the BondRx really does provide a reproducible, consistent, and fully automated workflow that works harmoniously with Opal. And Opal itself allows for the detection of upwards of 9 colors of interest on a single tissue section. So this flexible system together really does allow users to design their own panel, use their own validated antibodies, and then running on the Leica Biosystems BondRx, you're able to facilitate high throughput research in the clinical setting. So thank you all so much for your time. And if you have any additional questions, please feel free to reach out to our customer care at akoyabio.com. I'll hand it off to Traci now.
Thank you so much, Bethany. My name is Traci DeGeer, and I am the Director of Innovation for Leica Biosystems Advanced Staining Unit. And I'm going to speak to you just a little bit about the BondRx instrument that you heard Bethany talk about, that is used in conjunction with the Opal kits that are produced by Akoya and fall under the Phenoptics group. One of the things that is unique about the BondRx is that it is a fully dedicated research platform. And the thing that makes it so amenable to being used in research and in co-marketing with products like Akoya and the Opal is that the software has a lot of unique capabilities for openness. So one of the things that makes it so easy to use is that it gives researchers the ability to explore ideas. You can run lots of different types of testing based off of the openness of the software. You can accelerate testing programs because the 3 drawers on the instrument function almost like 3 separate stainers. And that allows you to not only run kits like the Opal, but also do some of your routine research work if you'd like to do that. And as you've seen with what Bethany's group at Akoya have been able to do, they were able to actually commercialize a very wonderful and unique idea with us. So we're going to talk just briefly about some of the unique features. The Explore Your Idea is one of the things that really attracts groups like Akoya and Leica together. And that's one of the things that makes our relationship so wonderful, is that we're able to automate so many different types of tests in the research lab, and that helps us advance the treatment of cancer and other diseases in the research world. So if you're looking to do research, then you can do wonderful assays like the Opal on the instrumentation. If you're looking to develop something on your own, there's always that unique spot where your test can fit in based on the things that you can do. And there is a long history of being able to run tissue-based assays on the Leica Bond platforms. Leica has been around for over 50 years in anatomic pathology. And so one of the things that is the hallmark of that is the tradition of quality instrumentation, microtomes all the way through. This is a little bit about the technology timeline of the Bond instrumentation itself. The Bond RX has not been around quite as long as some of the Bond platforms. It actually did not come into being until 2012. And that, if you look at the scheme of some of the instrumentation that's been on the market, is not very long. So the instrument was released in 2011, and then immediately there was this desire to start making the software into something very, very unique. The Akoya group was actually our second co-marketing partner that came on board with our open innovation team and has been a strong partner for us ever since. We started with them when they were still with PerkinElmer, and have built a very strong relationship with them all along. And that's allowed us to bring some of the wonderful testing that we've been able to bring to you through that unique partnership. If you look at all of the possibilities that you're allowed to do, or we enable as a team for you to do on the instrument, you can do multiplex fluorescence, you can do in situ hybridization, you can do immunohistochemistry, LNAs, mRNA. All of those things are possibilities within the instrumentation itself. Last year, one of the things that came about with the Bond RX was the release of the 6.0 software. That gave researchers greater capability in what can be accessed and done with the software itself. There are a lot of things that the software has always said you had a lot of freedom to explore those things. One of the goals of the team has always been to increase that capability. There's additional pre-staining capabilities now. There are additional customizations that you can make in order to control some of the things that we used to have to send field service representatives in to do for you. So you have that greater flexibility to do those things yourself now without having to wait on us. And that was one of the things that we were very excited to bring out. The pre-staining lets you adjust times and temperatures a little bit more. You can add and subtract alcohol steps. The tissues are not always created equally. That was one of the things that through the years and experiences the team came to realize. And so sometimes it can't be treated equally. And that gave greater optimization for our users to be able to adjust to those conditions within their own lab, or even to tissues that come in from other places. Because sometimes if you're working on research studies, you can't control what comes into the laboratory. And so this gave that added bit of flexibility to those pieces. There's also the ability to change incubation times and temperatures for antibodies, for rinse steps, all of those types of things, applications for probes and antibodies. All of those pieces have evolved over the time and the life of the instrumentation itself. One of the unique features that was really exciting for us was the dispense type. That's something we used to have to send either application specialists or engineers in to adjust for either our partners like Akoya or even our customers who work in the research laboratories every day. If there was something unique they wanted to do and it required either an open dispense or it required a different type of dispensing than what was installed when the instrument left the factory, we would have to send someone in to adjust that for them. the instrument allows the researcher to adjust that on their own now, so they can actually experiment a little bit more and change those things on their own. And that gives a little bit better adaptability for the instrumentation without them feeling constrained to wait on us to be able to get into their laboratory to make those adjustments for them. And then the staining customization, you can determine whatever type of incubations you need. You can change things for the washes. You can actually change things for a variety of multiplex protocols. And that allows for greater optimization on the part of the researcher. We'd like for the instrument to be able to bend to the will of the researcher rather than the researcher bending to the will of a piece of instrumentation. And that's always been the goal. With those goals, it allows people to accelerate their testing programs. And the reason that's such a wonderful thing is it gives you more opportunities to actually test more. If you have multiple groups working in the laboratory, then your instrument with the drawers can actually give you more opportunities to put more teams on the instrument. If you don't want to start all 3 drawers at the same time, then that works great. The instrument can be very quick, depending on what you're running. If you're running a 9-plex, then, as Bethany said, that will probably be an overnight run for you. We do have other assays that are overnight runs, but basic IHC can be very quick, depending on the needs of the laboratory itself. It's very efficient. One of the things that it will do is that depending on the version of the instrument that you have, it will tell you when fluids are low. You can do your visual management from across the laboratory and not have to sit and babysit the instrument. The openness of the containers, you can set up titration vials and let the instrument do your titrations for you. You don't have to come back and take care of that on your own. You can run open detection. There is added flexibility coming in the open detection this year. So there'll be some unique things coming down the pipe that we're hoping will be out by the end of the year, hopefully, if not by the first of next year. The visual management of the instrument is very nice. You can see everything on the front screen, which lets you from across the room while you're carrying on important tasks in your laboratory, be able to look at a glance and see what's going on with your instrument. And if there are things you want to add or remove or take care of, then you can do that seamlessly. The bulk fluids changed to allow them to be part of that visual management as well. And customers have found that they very much like those pieces. Now, I came from another company, and one of the things that we used to pick on quite a bit was the cover tiles. Now that I have joined Innovation, I have actually found that the cover tiles are a very unique piece of what we do. They provide a lot of safety and security to the tissues on the slides themselves and allow for some of the unique partners that we've been able to form in Innovation. They actually provide a lot of safety and security to everything that goes on to the slide, and that has enabled us to form some of the unique partnerships that we've been able to form. And as we move forward on the BondRx, you're going to see some very, very unique partnerships come out of the ability that we do use the cover tiles that we use. And they are a very wonderful piece of what we do. The Opal assay actually works excellent with the cover tiles that we have on there, and it helps us do a lot of long assays when you look at the overnight piece and get good staining with those and protect our tissues at the same time. So we have a lot of wonderful partners that we work with, and all of those have come about based off of the relationships that we were able to build with our first partners, which were ACD and the Akoya group. So if it hadn't been for them, then it would have been very difficult for us to move forward and build some of the partnerships that we've got today. So Akoya is a strong partner for us. Their technology has been a wonderful addition to the research world and continues to grow and expand across the globe and is a wonderful asset to the research world. As the microenvironment becomes more and more important to cancer research, it's very important for all of us to be able to see a little bit further into the tissue and the spatial relationships that we get between the cells. And this has helped drive both the research world and our own look into the microenvironment. As we're looking at multiplexing, you also see a lot of the CTC world going that same direction. You will see them multiplexing with fluorescence as well. And one of our partners is also in the CTC world. And you notice that they use many of the same type of colors that you will find from Akoya. These are different types of cells and not tissue-based. But the circulating tumor cell world also leans very heavily on the fluorescence. You do see a lot going on in the multiplexing world. This is another of our co-marketing partners. This is UltraView, and they also do multiplexing. And our other long-term partner that we have worked with for a very long time is ACD. And a lot of what we do in the innovation for the BondRx came up on the backs of both ACD and Akoya. And ACD is our mRNA partner. And they have launched several different types of products with us, just like Akoya has. And with the mRNA, they are our longest lived partner, and they actually were our first partner to go from the research world into the clinical space. So that's where the commercializer discovery piece comes from. One of the things as a researcher that is very unique is the fact that when you run on the BondRx, you use the same software that both Akoya and ACD and the other partners run on their instrumentation. So with the open detection kit and the ability to run the open software and create an assay in your laboratory that has the ability to change how people view medicine or how people treat cancer, those technologies that you use in your lab are the exact same that all of our partners use. So the things that make that unique are the ability to put whatever you need to put into those open detection vials, the ability to customize your protocols however you would like to customize them. But at the same time, you get the standardization of using a routine bulk fluid, routine image retrieval, and you get the run-to-run consistency that Bethany showed you when she went through her procedure that they tested for the Opal products. And that gives you a combination of flexibility, but the consistency that you would want if you were going to publish or if you were going to develop something on your own that you eventually wanted to show. So it's that combination of the two that makes the partnerships unique. So it creates a pathway from the instrument to partnerships like Akoya to eventually something that maybe someday we'll see in the clinic. And those are the things that all of us who work in cancer research are hoping to someday see. So that's the path that some of our partners have taken. And hopefully that maybe you will see in something that you do, but we're proud to call Akoya a partner. And I hope that you find value in what we do together. And if you have any questions at all, please reach out to either your sales rep at Leica or please reach out to Akoya if we can answer any questions at all for us. And let us know if there's anything at all that we can do to help you. Thank you so much for your time. And I'm going to hand the mic back for open questions.
Thank you, Traci and Bethany, for your presentations. And it is now time for our live Q&A portion of our webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. As a reminder, any questions that we are not able to answer live today and those submitted during the on-demand period, Bethany and Traci will be answering them via email to the address you provided at the time of registration. Okay, let's get started. Bethany, let's start with you. How about this question? How long does assay development typically take for Opal on the bond?
That's a really great question. So typically, we would say that with our support and following the steps that we provide, as well as what Leica provides, typically we would say that between determining antibody order and pairing, running through the monoplexes, and then transitioning to a multiplex, we say that for your first panel, it would be about a month. That can, of course, vary depending on your markers of interest as well, and if, you have variability within your staining pattern. However, we average about a month for a new panel.
Thank you, Bethany. Traci, let's pop over to you. How long does the 6-plex assay take on the Bond RX take from beginning to end?
So that's an excellent question. So if you look in the literature, the 6-plex assay plus the DAPI can take up to about 14 hours. And this is once the assay has been fully optimized. And that's why the recommendation is that if you're going to run the full assay, that you run it overnight.
Thank you, Traci. Bethany, let's come over to you. Can I dilute out my primaries since Opal amplifies signals so much?
Great question. So as mentioned in the presentation, we typically recommend that you try to titrate your Opals up or down, depending, on what signal you want to achieve. But sometimes when you transition from a tonsil monoplex to your tissue of your project monoplex, that, sometimes there can be discrepancies that titrating Opal can account for. So during this instance, we would recommend actually either titrating your primary antibody then, for example, like if it's CD8 staining, it'll be very strong and intense. So in that circumstance, you would want to perhaps dilute out more your primary antibody, but we would try to recommend trying the Opal titration first before taking a step back to dilute your primary.
Thank you, Bethany. Now we have some great questions coming in, and we have time for just a few more. Traci, do you always need to run a 6-plex assay on the Bond RX, or can you run up to a 6-plex depending on your needs as a researcher?
That's a great question. You do not always have to run a 6-plex. If you wanted to structure your assay so that you're running a 3-plex or a 4-plex, depending on your needs as a researcher, you can actually adapt that to your own needs. It is not necessarily that you have to always run a 6-plex per se. You can structure that order however you would like to do that, and then determine the best colors to suit your needs.
Thank you, Traci. And Bethany will wrap with this final question. When interrogating co-localized markers, does it matter what order I apply them in?
Yes, these questions are really great. So, for example, in the example I showed, CD3 and CD8, it does matter sometimes what order you apply them in, because in particular, if they're on the same cellular compartment, there could be differences in expression levels. And so we typically recommend that when you are interrogating co-localized markers, that you want to put the lower expressing markers first and then have the higher expressing ones afterwards. But really, what you want to do is you will want to look at the staining pattern expression and assess from there. If you realize that when you stain one way, say CD3 and then CD8, that you aren't getting consistent expression of one of the markers, try flipping them and seeing if that will make a difference. And typically it will, but for the most part, we haven't really seen too much issue with these. But once again, excellent question. And if anybody else would like more information on that, please feel free to reach out.
Thank you, Bethany, and thank you, Traci. I'd also like to thank our audience for joining us and for their interesting questions. And as a reminder, those questions we did not have time to answer today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. I'd like to once again thank Dr. Bethany Remeniuk and Traci DeGeer for their time today and their excellent presentation. We'd also like to thank LabRoots and our sponsor, Leica Biosystems, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we hope to see you again soon.